Okay. Hi, good evening, everyone. Thank you so much to all of you who have joined today's session. Today's discussion will be on biomaterial development and 3D bioprinting applications. I'm Abhishek Kalra. I lead the 3D technologies business at Altum Technologies. And I'm very excited to have with us today Dr. Kaushik Chatterjee from the Department of Material Science, Center for Biosystems Science and Engineering from the Indian Institute of Science in Bangalore. Uh, those of you who have been following the bioprinting space would already know him, but I'll still take a minute to introduce him. So Dr. Kaushik received his PhD in bioengineering from the Pennsylvania State University in USA and a MS in Material Science and Engineering from the University of Virginia, also in USA, and BE in Metallurgy from Bengal Engineering College in India. Post which he worked as a postdoctoral fellow jointly at the National Institute of Standards and Technology and the National Institutes of Health in USA. In 2011, he joined IISc Bangalore, where he is currently an associate professor at the Department of Materials Engineering and is associated with the Center for Biosystems Science and Engineering. And his research focuses mainly on materials development for biomedical applications. He's probably one of the more well-known people in the bioprinting space. We're very excited to have him here share some of his work. We also have on the call Ms. Aishwarya Shirur from Altem Technologies. Aishwarya is the technical leader for our life science products, working as an application scientist with the bioprinting division. So both of them have put together a very interesting deck, which will show you some of the interesting work happening in the bioprinting space. So I hand over the stage to Dr. Kaushik now to show us his presentation. Thank you so very much.
Thank you. Thank you, Mr. Abhishek, Ms. Aishwarya, and the entire Altum Technologies team for the invitation and opportunity to discuss some of the 3D printing and especially bioprinting work that we've been doing in the last, maybe couple of years now. And as you already heard, we are a laboratory based out of IISc in Bangalore, and we are a biomaterials lab, and we've been working with a variety of biomaterials uh, over about a decade now, a little more than a decade. And while we've used a variety of technologies, what has really been sort of a huge change in this field in the last few years has been the introduction of 3D printing, and that has really started to make a huge difference in the field. So before I start talking about the specifics of what we've been doing or what the opportunities are in this field, just let me remind you that when you talk about materials for biomedical application can mean a lot of things to a lot of people. There are applications starting from say biosensors, there's drug delivery, uh, and then a host of other things. In our group, we basically look at two major classes of applications. On the left, these are the conventional medical devices or implants that go inside the body. So we do a lot of work with titanium alloys or other metallic alloys that are used to prepare many of these conventional medical devices. Even though they're widely used in the clinic, there's certainly scope for improving them. And we look at how we can improve the performance of these materials that are used to make these devices. And in recent times, we've actually been using a lot of 3D metal printing to try and come up with enhanced properties and see how to improve the performance of these devices. But that's not something I'm going to talk about today. But what I will talk about is the work we've been doing in this tissue engineering space. And this schematic on the right tries to capture that idea. I'm guessing most people in the audience today would know of it. So I'll just summarize it quickly by saying that the idea is to be able to replace and regenerate damaged tissues and organs in the human body. And essentially, what you're talking about is to take cells either from the patient himself or herself or from a different donor. You expand the cells. To grow a tissue, you need a 3D construct in which you see the cells. And that is where the biomaterial component comes in. Typically, these are some biodegradable materials, and more often than not, these are biodegradable polymers, because most tissues in the human body are quite soft, and the biodegradable polymers work out quite well in terms of matching the properties of human tissues. So you seed the cells on these what are known as tissue scaffolds, and feed them with biomolecules like steroids or hormones and growth factors and other proteins and so on. The idea is to start or initiate the tissue formation, and then the cell seeded scaffold can go inside the body and help in regeneration of the particular tissue or organ of choice. Now, what we do, of course, is work on the choice of the biomaterial itself, and that again depends on the particular tissue that you're interested in. I will particularly highlight some of the work we've been trying to do in the context of bone tissue engineering, which can also be extended to some of the other tissues, and particularly how one can use 3D printing technology to address some of these challenges. Okay? So, before we get into that, I just wanted to talk briefly about this idea of 3D printing, what is also known as additive manufacturing. So, conventionally, if you think about any sort of manufacturing, it comes down to uh, two ways of making or manufacturing a product. The first one is this formative manufacturing, where you think of casting. So you think of a mold, you fill it with, say, liquid metal, liquid polymer, liquid ceramic, or powder of ceramic, and then you fuse it or you let this liquid metal cool, and you get a part of interest. So this is formative manufacturing. In subtractive manufacturing, you start with a block of material and then you machine it to the part of your interest. So where you remove the material, and therefore it's subtractive manufacturing. What has come up in a big way in recent times is this idea of additive manufacturing. This is basically a process of joining materials to make objects from 3D model data, layer by layer, in contrast to subtractive, where you actually remove material, right? So now, when you do this, there are various ways of doing it. One is to use what are often known as powder bed technology, where you just lay down powders layer by layer, and you use an energy source like laser or electron beam and so on to fuse the material. Or in many other cases, you use a head, a movable head, to typically deposit uh, a material of interest from that nozzle, and that in particular is known as 3D printing. So in some sense, 3D printing would not include technologies where you just rastering a laser without depositing material from a nozzle head. They would all come under additive manufacturing. So 3D printing is part of additive, but some technologies of additive are not 3D printing. Having said that, I mean, that's more of a technical sort of nitpicking that one does, but that's important to recognize it. Nevertheless, I think the point here is additive and 3D printing are widely used interchangeably, and they are certainly very contemporary topics of research and of much interest to uh, both the research community and also to industry for a variety of applications. So if this is all that great, the question is, why not do this 3D printing for everything, right? Why not just stop doing formative and subtractive manufacturing? Why would you do 3D? I mean, why not adopt 3D printing for everything? And the answer lies somewhere here in this plot. You can see that if you have, I mean, you're trying to make very few parts, then you're making the mold to do formative manufacturing would be quite expensive per part. However, you're using that same mold to do mass production of hundreds. Better is if you can do thousands and millions of pieces, then the cost per part would actually drop very, very fast. So for mass production, you're still better off using conventional manufacturing like formative manufacturing, and machining is somewhere in between, where you would actually get some benefit per part. However, when it comes to additive or 3D printing technology, it's pretty much the same per sample. So you really don't get any benefit in terms of increased number of parts, and the cost per part will remain the same. So where you do get the benefit is you're trying to make complex parts, so complex geometries, which will be difficult to prepare by other kinds of manufacturing. And also, if you're trying to make only a few number of parts, then certainly you get the benefit of the cost. And so that is where the advantage lies when it comes to 3D printing or additive manufacturing. So when it comes to polymers for biomedical applications, you can imagine the wide variety of uses that people have demonstrated over the years. There are, of course, the non-degradable polymers, but there are also, the biodegradable polymers, and we are actually going to talk more about the biodegradable ones because they can be used as tissue scaffolds. But polymers in general have been used over several decades and maybe more than a century now in a variety of biomedical applications, starting from making parts of devices, also for drug delivery, for gene delivery, for tissue engineering, biosensors, and probably many other applications. Okay? So polymers are indeed very attractive class of materials that are used in variety of biomedical applications. Okay? So specifically when it comes to polymers that are used, or biomedical polymers that are used in conjunction with 3D printing, where it has really taken off in a big way and there is actual sort of both interest, both in terms of commercial applications and, of course, research, is this idea of making patient specific devices. Now, this device could be, of course, a conventional medical device, or it could be a tissue scaffold. But the idea is that you don't mass produce these implants, but make them very specific to a particular patient. And the principle is captured in this image. So basically, you take a patient, let's say somebody needs a part of their jawbone replaced. So you can imagine your jawbone and my jawbone will obviously not have the same shape and size. And also, the defect in your jawbone might have a very different geometry than the defect in my jawbone. So the idea then is to scan that defect site, go to a computer or a software where you can recreate the model of that defect site, prepare a scaffold or an implant that would be appropriate to fit that defect, use a 3D printer to make an appropriate kind of scaffold or medical device, and then prepare finally fabricate the part of interest using 3D printing technology. Since you're talking about low volumes, because you only need one or two parts per patient, if you remember the costing per part, this is where 3D printing becomes very attractive. So this is where 3D printing has taken up, is coming up in a big way. Okay? So then, and of course, aside from that, there are a variety of other applications also where it's actually starting to come up in a big way, and that includes, the idea of using this of using 3D printing to make mimics of human tissues where you would like to do some surgery. So if you think of uh, a patient who is going to undergo some sort of a complex surgical procedure of their heart, it's difficult for the surgeon to prepare for the surgery in advance until they open up the patient and see what the tissue looks like. Whereas if you could scan that particular tissue and then use 3D printing to create a model, which is basically a surgical mimic of that tissue of interest, and then you can fabricate that, you can give it to the surgeon, they can look at it in 3D, they can hold it in their hand. Many of these polymers that are used for it have the same kind of mechanical feel that you would see for human tissues. They can visualize it in their own hands, plan what the surgical procedure they will do, and accordingly they will, be better prepared for surgery than just opening it up, opening up the patient at the time of surgery and trying to figure it out. So this is a big area where people are looking at 3D printing or surgical mimics and using uh, usually some sort of a rubbery or elastomeric polymers for these kind of applications. Uh, Another area that is coming up with respect to 3D printing is this idea of personalized drugs. So here again, much like the implants, where each of us has a different defect site that we need to fill. Similarly, in the case of drugs, maybe how you respond to drugs might be different from how I respond to it, or the kind of dosage that you require might be different from mine. So the idea is, can we actually prepare customized, say, pills or tablets, uh, and including, you have multi layers, so that maybe one drug comes out first, the second drug comes out later, So all of these designer drugs could be prepared by 3D printing. So you again design it on the software, you can fabricate it. Before that, you start with the diagnosis, the doctor writes a prescription, and then in this printed device, the printed drug, it would be personalized for that particular patient to have a very custom uh, outcome or therapeutic effect on each person, which would be different for each one of us, of course. Okay? And here again, the choice, not just the amount of drug, but also if you coat it with various polymers, then you can control the release profile, which is where the printing of polymers with the drug becomes an interesting area to pursue. Okay? So, what I want to do today in terms of showcasing some of our own activities, I want to talk about this 3D printed polymer scaffolds. And of course, the overall or the long term goal is to have patient specific scaffolds. But right now, it's the work that we've been doing essentially is to see what one can do in terms of choice of material, and also how to process it in the context of 3D printing so that you can get scaffolds of interest that are usable while you maintain good bioactivity, and then develop this further for biomedical applications, in particular for tissue engineering. So, just to reiterate this idea that for patient specific implants, one would scan the defect site, uh, generate the 3D model in the software, which would then be connected to a printer to prepare a scaffold of interest. And you know, uh, one could do one of two things: print along with cells, which would be bioprinting, or print the scaffold first and then add cells from the top to colonize the scaffold, which would be 3D printing of polymers, resorbable polymers, and then seeding cells on top of them for generating the tissue. So the first technique that I want to talk about is this extrusion-based technology. This is, one of the names for this is fused filament fabrication, or FFF. These are this is a very simple technology in the sense that technically it's simple, it's affordable in the sense that the printers are not very expensive, uh, and also at the moment you can find several suppliers who can supply printers for you in various price ranges. The simple ones can actually be only 10-20,000 rupees also. Of course, they don't necessarily have the best sort of technology in terms of precision and rates and versatility and so on. But nevertheless, they are good to get started. And then the other downside, of course, is you don't get very good resolution. The feature sizes are fairly large, and also the surface tends to be rough with a lot of porosity in it, so it's difficult to make a dense part. However, that is an advantage in the case of tissue scaffolds where you are trying to make a porous structure. So it's actually quite convenient to prepare these simple porous geometries for tissue scaffolds. And the other limitation, of course, is you can't incorporate cells of biomolecules because they will get denatured. And that's because the way it works is you've got a spool of material of the polymer of interest. It's typically like a thermoplastic, which will melt when you heat it. And so when you pull this filament it through rollers and you feed it into an extruder head, there are heaters to melt the polymer and push it out and lay it down layer by layer. So the head can move in X and Y, and the stage can move down in Z axis, and that way you can build it up. And polymers like polylactic acid are widely used with this kind of technology, and that's also a biodegradable polymer used in biomedical applications. So I'll show you some work we started doing this several years ago, and this is a former postdoctoral fellow in the lab who started doing it with a very simple 3D printer that we had, sort of acquired commercially, and we set it up in the lab. And you can see here that there is essentially a printer head, and we have given a design. So you can see the figure on the left. This is the model that we prepared in the CAD/CAM software, and these are struts that are few hundred microns in diameter with a spacing of about 400 to 500 microns. And this is what we feed into the computer, and then the printer head prints it. So you can see it in the yellow box here. So this scaffold has been printed. The idea is to use what is known as a 0-90-0 sort of arrangement, where the first layer is at, let's say, at 0 degrees, and the next layer is at 90 degrees on top of it, and the next layer above that is again parallel to the first layer. And these are made out of polylactic acid. This is a biodegradable polymer, which is actually well established for use in a variety of biomedical applications, including in tissue engineering. So this is a non-toxic polymer, which works well for many of these applications. However, what it lacks is the ability to promote bone formation. So while it's non-toxic, if you seed cells, they will grow on it, but they don't necessarily turn into bone, which is what we would like the cells to do in our case, because we're interested in bone tissue engineering. So what we set out to do was to modify the surface such that it would be more conducive for bone tissue engineering. So if you look at the composition of bone itself, and if here's a schematic trying to explain what bone is like. So you can see here that the bone is made out of composite material. So there is a protein, which is your polymer, and then along with the polymer there is the ceramic component, this calcium phosphate, and specifically in the form of hydroxyapatite. And of course, this polymer is your collagen fibrils there. So the bone tissue itself is a composite material, which is polymer plus ceramic. And this particular ceramic is hydroxyapatite. And this hydroxyapatite has been widely used in a variety of applications, starting from bone fillers and tissue scaffolds for bone, to coating implants for orthopedic and dental applications, and so on, and also as nanoparticles for drug delivery. So it's actually a well-studied material. So when it came to modifying our scaffold, we thought, why not modify it with hydroxyapatite so that you can actually get, make it more sort of conducive for bone formation. So that's what we tried to do. So what we did was to take our scaffold and put it in what is known as simulated body fluid, or a supersaturated solution of calcium and phosphate ions, hoping that they would actually deposit on these scaffolds. That really didn't work, because what we found is even after several days of putting it in the solution, nothing was actually getting deposited. So it wasn't very good, or the substrate wasn't great for depositing calcium phosphate on it. So we realized we need to modify that. So what we did is basically start with this polylactic acid. This was dipped in sodium hydroxide solution so that you can actually generate some OH groups on the surface. And then we conjugated low molecular weight polyethylene amine to it. Okay? And what that did was to essentially put some amine groups on the surface of the polylactic acid. And on top of it, we conjugated citric acid. The amine and the citric acid groups were conjugated, so that now you had a surface that was decorated with citric acid. Now, why citric acid? Because naturally it's present in small amounts in bone tissues, and its role is to help in the nucleation of calcium phosphate. So when we took the citric acid decorated scaffold and we put that in SBF, what we found was that there is hydroxyapatite getting deposited on the surface of this scaffold. And so what we did next was to study what the surface was of the as printed scaffold, and also after each of these steps of modification, all the way to the last step of hydroxyapatite deposition. So what you can see here is that this is the porous structure of the scaffold at various magnification, increasing magnification. The last row is for zoomed in onto an individual strut of the scaffold. And here on the last bottom corner, you can see the white specks, which is the calcium phosphate that's been deposited on the surface. So then what we did was to take mesenchymal stem cells from the bone marrow. So all of our bone marrows have these stem cells, and their role is to turn into bone-like cells. And we had isolated them. Actually, we procured it from someone who had isolated them from human bone marrow, and we seeded the cells on the scaffold, either the as printed scaffold itself or the scaffold that had been treated with the NaOH to introduce OH groups and make it more hydrophilic, and the final hydroxyapatite coated scaffold. And then what we did was to look at how the cells were growing. So, and then here we've zoomed into an individual's strut of that scaffold. And you can see one day after seeding the cells, they look more or less the same, some cells on the struts of the scaffold. However, by day 7, you can see that the cells are growing on the as printed scaffold, but not so well. Whereas when we have hydroxyapatite on it, you can see that the scaffold is completely covered with these stem cells. And you can quantify them with various assays. And what this tells you is that, yes, indeed, having the hydroxyapatite coating seems to help compared to the as printed scaffold. However, the other important thing, aside from growth of the cells, is their ability of the stem cells to turn into bone-like cells. There are two ways to look at it. Well, there are several ways, but two ways we did was, one was to look at alkaline phosphatase. That's an enzyme that there's more of that enzyme when the cells turn into bone-like cells. And the other is to look for calcium phosphate deposits that the cells will secrete, and that you can measure by staining it with a dye that is red in color. So you can see here that there's more alkaline phosphatase expression on scaffolds that had the hydroxyapatite coating compared to all these other scaffolds which did not have that or only had these other coatings. And also, if you look at the amount of calcium phosphate that's deposited, you can see here that the one with hydroxyapatite coating has the most amount of mineral deposit on it. It looks the dark red in color. Okay, and that's quantified here in this bar graph. So what we are saying is you can print a scaffold using polylactic acid, maybe a patient-specific scaffold. However, to make it more conducive for bone growth, one could modify it with hydroxyapatite and then use it for bone tissue engineering. It will just become more conducive or more friendly for these applications. Okay. The other strategy that we took, and this is the work of a current PhD student who's doing this, is instead of looking at hydroxyapatite, which of course many people have done over the years, not for 3D printed, but many other kinds of scaffolds, is to look at what are known as redox modulating or ROS scavenging ceramics. In particular, we were looking at Ceria nanoparticles. Okay, so cerium is an element in the lanthanide series, and when it forms oxide, it exhibits some very unique properties that it can exist in both +4 and +3 states, and it can switch between the two, and that finds applications for catalysts, for sensors, and so on. And even in biomedical applications, people have taken Ceria nanoparticles and they have put that into the body, at least in animal models, and shown that it can have antioxidant effects, it can be pro-angiogenic, and that is mediated by the ability to scavenge ROS or a reactive oxygen species in the body. So the idea was to see whether if you took a scaffold and modify it with Ceria, could it help in bone formation? Because the idea is that when there is new bone formation, there is a lot of ROS that is generated, and could you modulate that with nanoceria coated scaffolds? So what we've done here is take, again, the same idea is to take a scaffold of polylactic acid, modify it with citric acid, and then put it in a suspension of cerium oxide, and we get scaffolds that are decorated with cerium oxide particles, as you can see here. And then what we did was we checked it with XPS, or X-ray photoelectron spectroscopy, and what we found here is that there are both C3+, CE3+, and 4+ states. There's a mix of the two, which is great because that means the cerium can switch between the two states and help in modulating reactive oxygen species. When we looked at the surface roughness, it wasn't very different for the Ceria coated versus the one that had been OH treated. So the surface roughness wasn't different, but what was different was there were cerium oxide particles on these. So next what we did was to seed cells on them, again the stem cells, and see what happens. And what we found here is that if you seed cells on these and you look at their ability to grow from day 1 to day 7, you can see that this number of cells is increasing on both the OH treated scaffolds and finally the Ceria modified scaffolds. And you can see that in the bar graph that there's not much of a difference. Both the scaffolds are good at supporting cell growth. However, if you look at the ability of these scaffolds to scavenge ROS, that was very different because the one with the Ceria could do it, whereas the other one obviously doesn't do it. So here what we've done is we've added a dye which will measure how much ROS is there. If there's more ROS, it will look more fluorescent, and by looking at the fluorescent signal, we can tell if there is reduced ROS quantification or not. Uh, Next, what we did was do these cells to look at osteogenesis or bone formation. So here again, not surprisingly, if you find that the cerium oxide helps in increased alkaline phosphatase and increased production of calcium phosphate, both at 14 and 21 days in culture. And you can see here that the one with Ceria has more red dye. It says there's more calcium phosphate than the one without cerium oxide. And that is because the ability of these scaffolds to modulate ROS, which the one without the Ceria cannot do. And in fact, ROS has another consequence, which is if you actually grow bacteria on these scaffolds, the ones with Ceria can help in reducing the attachment and growth of bacteria by some more than one log order. Okay, so this is important because many of these implants are prone to bacterial colonization and infections. So if you can reduce bacterial infection, that actually helps in improving the bioactivity of the scaffold, and that is actually desired. And lastly, always the question in our mind is, what is happening in the scaffolds or on the scaffolds for the cells that leads to these changes in osteogenesis. So we collected RNA from the scaffolds, we did next gen sequencing, and then we did gene ontology to look at the changes in gene expression. And to summarize, basically we found that in the presence or absence of Ceria on the scaffold, there are several genes that are differentially regulated. And also, if you start looking at what pathways they affect, is primarily the cytokine receptor, and the chemokine related pathways, which again is not surprising because they are involved in, they are known to affect osteogenesis, and because of the changes in ROS levels in the scaffold with the Ceria, these pathways are getting affected, which in turn are known to affect bone formation. Okay. So with that, let me switch to one other kind of 3D printing, which or a subset of 3D printing with polymers, which is called bioprinting. So here we work with hydrogels. Okay, so these are basically uh, cross-linked water-swollen polymer networks, and when you cross-link the material or the polymer pre-polymer, you can entrap cells in them, and if you take a degradable gel system, then the cells would continue to grow and form the tissue while the polymer network would fall apart. Now, people have been trying to do 3D printing in the presence of cells. So, earlier what I showed you was you print a scaffold or you print the polymer first and then you add cells on top. Uh, However, the distribution of cells can be a problem. Also, it will be difficult to spatially pattern the cells in different regions of the scaffold, which you could do with 3D bioprinting technology. So here there are four kinds or four subsets of this technology. One of them is inkjet printing. That is where you have a solution or a gel rather, where you can have cells encapsulated. There's an actuator which works on thermal or piezoelectric effect, so that little drops of this, what is known as a bioink, would come out of this nozzle and get deposited layer by layer. Or you can have the same gel put on a slide where you shine light, and you can again get these drops of gels to get deposited on the surface. But what is by far the most popular way of doing 3D printing is extrusion-based bioprinting, where you have a printer head, typically some sort of pneumatic or screw system, where you take the gel with the cells and you push at the one end so that through the nozzle the gel will come out, and it will get laid down layer by layer, and you can build up a cell-seated scaffold uh, as your 3D printed part. Also, what the other technology that is coming up in a big way is the stereolithography based printing or digital light based processing technology. So here there is actually nothing that comes out of a nozzle. Here you have a vat or a bath of pre-polymer solution which is sensitive to light, and you shine a light source at particular locations. When you do that, in that particular location, the polymer gets, I mean, these are photosensitive polymers. They get cross-linked. The cells are trapped in the gel, and you will end up creating a cross-linked gel in this bath itself. So I'll give you some examples of this particular technology of DLP printing, which we've been trying to use in the last year and a half or so. So again, before we get into that, just to emphasize that each of these techniques comes with its own pros and cons. When it comes to any of these extrusion-based technologies, there is a limitation of how viscous the solution can be when you can extrude it or use it for an inkjet technology. So you can't go up very high. In stereolithography, you don't worry about it. In terms of cell densities, in inkjet, it is fairly low. When it comes to some of these other technologies, this is not a constraint. Finally, also there are differences in speed and cost, but certainly all of these have their own value. And this is certainly, I mean, one can choose their printing technology based on what they have in mind in terms of the application, the resolution, the kind of cells, the kind of polymers, and so on. Okay. So next is, just to give you a couple of examples of work that we've been involved in using this DLP or digital light-based printing technology. And like I said, these utilize photopolymerizable polymers, which means then that even biopolymers have to be made photosensitive. And what we do here is we add acrylate groups. So in this particular work done by a former postdoctoral fellow, we've actually taken silk fibroin and we extract it first from silk cocoons. So this is a well-established process. So you start with the silk cocoons, you do this degumming, then you dissolve it in lithium bromide solution, you dialyze it, and you lyophilize it to collect the powder. And then what we do is react it with glycidyl methacrylate so that you can introduce an acrylate group on this on the silk. What that does is it becomes photosensitive. So we use this selling machine Luminex for our work, where we have a vat where we take this acrylated silk, we dissolve it in an aqueous buffer or in media, and then we have a photo initiator in it, and then you shine the light right at the plane in which there is a stage, so that the polymer gets cured there and it gets sort of stuck to the stage. And then you lift the stage up a little bit, and again the next layer of pre-polymer is cross-linked. The cells are suspended in the solution itself. So as we build this up layer by layer, we are able to build up a scaffold of interest. So we've looked at a variety of properties, starting from degradation rates and mechanical properties and so on. So if you vary the polymer concentration or the silk fibroin concentration, you can get various kind of properties. And then you can make a variety of complex structures. So the first row and the top row and the third row are basically the designs that we feed into the computer, and the second and the fourth row are the printed the corresponding printed scaffolds that we've created. So you can see some interesting structures here. There's a channel within a hydrogel of silk fibroin. So here again is another set of channels. These are cortical and compact, cancellous and compact bone structures that we've created and printed using silk fibroin. Here again, there's lung or blood vessel-like structures that you can create. These are purely made out of silk fibroin that has been made photosensitive. And in principle, you can encapsulate cells. But since we were interested in bone tissue engineering, what we did was to take MC3T3 pre-osteoblasts, and we encapsulated them in scaffolds or these hydrogels, which were of 10, 15, and 20% polymer, and which meant that the stiffness varied from about 10 kilopascals to about 100 kilopascals. And what you find here is that the cells were viable in all of them. However, the cells seem to grow the best in the 15% silk fibroin gel of intermediate stiffness, not the very soft gels and not the very stiff gels. It was the intermediate stiffness, about 40 kilopascals, where these cells were growing the best. And in fact, the cells were actually spreading out a lot in these hydrogels over time. And then when we looked at the ability of these pre-osteoblasts to turn into more mature osteoblasts and osteocytes, what we found is in that 15% gel, initially the cells are more compact, then they start spreading out a lot, and finally they become very, spread out, much like osteocytes, and that happens spontaneously as you culture them from day 1 to 4 to 7 and so on. And if you look at their ability to deposit calcium phosphate, which is the ability to form bone, then you find that it increases from day 7 to day 14. Further, if you add in soluble factors like osteogenic media, that is actually more intense, which we believe arises because the silk fibroin itself is a good matrix for the bone cells to grow. And by tuning the stiffness to 15% silk fibroin, we found that that was the optimal condition for the bioprinted scaffolds where the bone cells can grow well. In another approach, and this is work of another postdoctoral fellow, instead of using a protein-based polymer, we used a carbohydrate or a polysaccharide-based polymer. Here again, we modified it so that it's got an acrylate group, and we've done all the chemical characterization that yes, indeed, we have introduced the acrylate group. And then what we do is we can add a photo initiator in it, and then we do again this bioprinting. So you can see these bioprinted scaffolds. We can make various kind of structures. You can make a square grid. You can make honeycomb structures. You can also make spikes. You can make stent like this knit or a cage like structure, much like a stent. So these variety of structures is possible to do with this DLP-based printing. We can vary the concentration of the polysaccharide from 1 to 5%. That, of course, means that at higher concentration, the pore size becomes smaller and also the stiffness starts to increase. Nevertheless, all of these scaffolds are great for encapsulating cells. So we have taken NIH3T3 cells and we have cultured them in these scaffolds. Particularly, I'm showing you data for the 2 weight percent pre-polymer solution that was used to prepare the scaffold. And you can see here that from day 2 to 5, all the way up to 30 days, the number of cells is growing. And you can see the images here. These are showing cells that are completely populating the scaffold and demonstrating how that this is a great sort of matrix for the cells to grow. Okay, and I should emphasize that one has to play with the the printing parameters in terms of the laser power and the time required for curing each layer. All of those, of course, have to be optimized. If you under-cure it, then the scaffold will fall apart too quickly. If you over-cure it, sometimes that can harm the cells. So one has to optimize the printing parameters so that you can get the best bioprinting conditions where you get stable scaffolds, but the cell viability and growth is not compromised. Okay? So just to summarize then, hopefully I've convinced you about the utility of 3D printing and how one can make a variety of scaffolds, either out of thermoplastics or out of hydrogels, and particularly with DLP-based bioprinting. And of course, these are just a few examples, a lot more happening that you can track, in the scientific literature or even technology development happening in companies, variety of materials that are coming up, new printing technologies, new printers, a lot of excitement in this field. People believe that this is one of the most upcoming technologies in the world with very large projected market values. This is just from one market survey talking of billions of dollars worth of market world over, including in India. And you can see here that there are variety of materials all the way from metals and ceramics to polymers, both plastics, hydrogels, resins, and then variety of challenges that one has to overcome. But one could, in principle, use these 3D printed parts either as implants or for tissue engineering to address a variety of clinical needs. Uh, and let me just end my presentation by thanking a lot of people. Uh, this is a list of the current group of people working in the lab: PhD students, postdoctoral fellows, uh, master's students, undergrad students, project fellows who work in the group, large number of former students who also contributed to these efforts, and several collaborators within IISc and also elsewhere within India and around the world who help us, including a lot of clinicians now, who are practicing clinicians who see patients, who tell us what the challenges are that they face and how one could probably use some of this technology to address patient care. And lastly, of course, all of this wouldn't be possible without all the funding, and be grateful for all the funding that we've received from various agencies. With that, let me end and I'll be happy to answer any questions.
Second, yeah. Thank you so much, Dr. Chatterjee, for giving us so much time and giving us an informative session on details of how you've developed several materials and how the team has worked together. So, following this, I will be showing some questions as asked by participants on this series, and then we can end the session. So, I'll just go on picking the questions and reading them out for you. Okay, so there's one question which is, how is the cell viability beyond 7 days of study of the cell seeded scaffolds? This is specifically for the Nanoceria enhanced stem cells.
Yeah, so actually, I mean, for most of these materials, we found that the cell growth is, and I mean the viability and the growth are all great. Uh, I think I mean, as long as you start with polymers that are non-toxic, and something like polylactic acid, of course, is non-toxic. It's widely used in biomedical applications, so the cell growth is fine. Nanoceria as a material itself is also non-toxic as long as you keep the concentration low. And since we just modified the surface of the scaffold, it was not toxic, and it also supported the cells. What can happen though is the roughness of the scaffold, the wettability of the scaffold. Those also affect many of these. May not be harmful to the cells, as in may not be toxic, but if they are not terribly favorable, then the cell growth slows down, and that is where a lot of this, sort of material selection and modification comes in to optimize that, so that you get the best growth of cells uh, on these scaffolds.
All right, thank you. I will go to the next question. Um, yeah, so why were silk fibron based inks created? And what feature of silk help you decide on using that for DLP based prints or any other forms of printing?
Right. So silk is in recent times, I would say last maybe 2 decades, has been studied quite a lot as a biomaterial, and it's primarily because it's essentially a structural protein. If you think about the cocoon, it's quite sturdy, and of course, even when you extract silk fibroin from cocoons, you do make it into textiles and variety of other applications, including patches for wound healing and variety of other things. So, and in recent times, people have been processing silk fibroin into various porous structures, fibrous mats, and people have started doing 3D printing of silk as well. It's just that most of that work is in the domain of extrusion based printing. There's very little, I mean, really only a handful of studies I know of where people have tried this DLP based printing that we were trying. And particularly, why do it for bone? And that's because there are studies which say that the peptide sequence in silk is very favorable for the precursors of bone cells to turn into more mature bone cells. So inherently as a material, it's very it's a good choice for bone tissue engineering, and also it then can be appropriately modified for extrusion versus DLP printing. So in this case, we have to modify it with an acrylate group so that it's photosensitive, and so that's what our interest was in trying to establish that technology.
All right, thank you. So we'll probably take two, three more questions and then we can end the session if that's okay.
Sure.
Yes. So, yeah. So, what is the proportion of the polymer ceramic, ratios added to work with bone printing?
Right. So if you're talking about, I mean, okay, so let me put it this way. If you're talking about the work we did with ceramics, there we were just modifying the surface. So we weren't really mixing the ceramic with the polymer, um, which you could do also. I mean, that certainly is a possibility. But what we did was to take I mean, your polymers that are used as biomaterials. In this particular case, we're looking at polylactic acid, and then we modified the surface. The amount of ceramic is very, very little actually, because just a very tiny coating or a very thin coating of nanomaterials. In fact, it's small enough that it's difficult for us to actually quantify it, because bulk of it is really the polymer. Having said that, you could do it differently where you could mix in polymer and ceramic together. But one has to bear in mind that if you're doing extrusion-based technology, either extrusion bioprinting or your this extrusion of thermoplastics, the moment you add ceramics, it'll become more and more viscous, and then you need more sort of force or stress has to be applied, like more pressures will be needed to extrude it because it will be a more viscous material. So whether your bioprinter or 3D printer can support it is one question. And if you do it, say if you take, say, a hydrogel, let's say we take a silk fibroin, I add in hydroxyapatite particles in it, and then I try to extrude it. Now you'll have to apply a lot more sort of force or air pressure so that it can get extruded. And the downside is then it can affect cell viability. So that can be a challenge, which one has to sort out.
Okay, all right. So, in addition to this, since you mentioned about HAP, I had a question. So, I think somewhere you have mentioned about HAP coating.
Yes, basically we had uh, printed the structure first, and then we modified it so that we just the surface was coated with HAP. Because see, it's actually easy to like for the filament based technology that we were using, it was easy to to sort of buy or source just filaments of polylactic acid. However, if you want to, let's say, add in hydroxyapatite, you would have to mix polylactic acid with hydroxyapatite first, and then figure out a way of making it into a filament to be fed into your printer. And then you need the right sort of thickness of the wire or the filament. That again, one more step for whoever is doing it to get the right thickness and so on, which if you can do it, it's in principle possible. But then that might be a limitation for people who are trying it in their own labs, that they, if you want to mix it in the bulk of the polymer, you have to figure out how you're going to make a filament of that material that you can use as a feed source for printing.
So, when you said HAP coating, was there a specific way you coated it, or you just like immersed the PLA into a solution, or sprayed it on, or
Yeah. No, so we tried the usual way of putting it in simulated body fluid, but that didn't seem to work so well, which is why we did all of those additional steps of first putting citric acid on it and then
okay,
immersing it in SBF, because only then we could get at least some amount of hydroxyapatite getting deposited. Otherwise, HAP didn't seem to get deposit. I mean, did not deposit on the polylactic acid surface itself.
all right, all right. So one more question. So, what is the alternative materials you would consider if you were not selecting PLA as a material for printing?
No, I think any of those other thermoplastics, like in that family, I mean, anything very close to PLA, like polycaprolactone and all, are also printable. However, I think polylactic acid happens to be the most, sort of compatible or friendly as far as 3D printing, especially the filament technology, is concerned. Also, it depends on your printer, because some of the printers use a filament, others could just have like a syringe where you can feed in powder or pellets of the polymer, and that opens up more possibilities, because many polymers can't be made into filaments, or not as easy to make filaments like you can do for PLA. So if you use a printer, again, a thermoplastic printer head, but the feeder is, or this, I mean, you can feed in powder or pellets, then that opens up more opportunities for you.
So there are just two more questions, if you're okay with that.
Sure. Okay.
We can, yeah. So the question is, how is the material properties post printing with silk based DLP prints?
Yeah, so I did not have the time to go into all of it. It's available now. It's a published paper. You can have a look. So we, I mean, we had optimized it so that it wouldn't, degrade too fast, or while maintaining cell viability. So we were, I mean, I would encourage you to have a look in terms of mechanical properties and so on. We have done the characterization. I would encourage you to have a look at this paper. Uh, it's also there on our lab's website. So it does share all the details about uh, the, the the mechanical properties, the degradation rate. All of those details are available in this paper.
All right, thank you, sir. So all right, so I have another last question from here, which is, does structural stability uh, maintained post HA printing with bioprinting also challenging to tailor the rheology?
Yes. So here, I think, I mean, okay, I should mention that in our case, we haven't done bioprinting with HA, because the work I showed you was, printing PLA and then putting HA on it. So that wouldn't, and then seeding the cells on it. So that technically isn't bioprinting, it's 3D printing, but the properties of the scaffold didn't change much, because I mean, the bulk of the polymer wasn't affected. It was only the surface that was getting affected. If the talking about rheology, so that didn't matter, because I mean, it's post printing that you're doing things. However, let's say if you talk about taking a gel, mixing in hydroxyapatite particles in it, and then you try to extrude it, there certainly your rheological properties will change, and you would have to take that into account, because most likely you're going to need higher pressures to squeeze out your gel. It'll become much more viscous, and like I said, that you might hit the limit of what your printer is able to support. Even if that wasn't a concern, what would be a concern is whether you're applying so much stress on your cells that you start compromising on the viability of the cells. So, but that would depend on, maybe you could reduce the polymer concentration a little bit while adding a small amount of hydroxyapatite and try that.
All right, thank you, sir. So I think we are towards the end of all the questions. Thank you so much for answering these questions. Thank you, Dr. Chatterjee, for taking out time and coming on board with us to present to this set of audience. And on the other hand, Altum Technologies, thanks you a lot, and we hope to see more of you in our maybe our next sessions, or we plan to work together or something. Apart from that, thank you everybody else on board who's watching this. Um, these videos are available on YouTube, on LinkedIn, and you're free to ask any questions regarding these presentations. Putting forward to Altum, and we can probably get in touch with Sir on the answers. So thank you so much. Thank you everyone, and thank you so much, Sir, for your time. We hope to see more of you. So I'll end the session right now.
Okay, thank you again for the invitation.
Yeah, thank you, sir.